Having staged the reaction all four ways, once with DDA, once with DDT, once with DDG, and once with DDC, we have four sets of DNA chains. One consists of chains ending in DDA, one with chains ending with DDT, and so on. Now, if we could only sort all these many chains according to their respective slightly varying lengths, we could infer the sequence. How? A moment, please. First, let's see how we could do the sorting. We can place all the DNA fragments on a plate full of a special gel and place the plate of gel in an electric field. In the pull of the electric field, the DNA molecules will be forced to migrate through the gel, and the speed with which a particular mini-chain will travel is a function of its size. Short chains travel faster than long ones. Within a fixed interval of time, the smallest mini-chain, in our case a simple DDC, will travel furthest. The next smallest, C-DDC, will travel a slightly shorter distance. And the next one, CC-DDG, a slightly shorter one still. Now Sanger's trick should be clear. By reading off the relative positions of all these mini chains after a timed race through our gel, we can infer the sequence of our piece of DNA. First is a C, then another C, then a G, and so on. In 1980, Sanger shared the Nobel Prize in Chemistry with Gilbert and with Paul Berg, who was recognized for his contribution to the development of the recombinant DNA technologies. Inexplicably, neither Stanley Cohen nor Herb Boyer has been so honored. For Sanger, this was his second Nobel. He had received the Chemistry Prize in 1958 for inventing the method by which proteins are sequenced. That is, by which their amino acid sequence is determined, and applying it to human insulin. But there is absolutely no relation between Sanger's method for protein sequencing and the one he devised for sequencing DNA. Neither technically nor imaginatively did the one give rise to the other. He invented both from scratch, and should perhaps be regarded as the presiding technical genius of the early history of molecular biology. Sanger is not what you might expect of a double Nobel laureate. Born to a Quaker family, he became a socialist and was a conscientious objector during the Second World War. More improbably, he does not advertise his achievements, preferring to keep the evidence of his Nobel honors in storage. Well, you get a nice gold medal, which is in the bank, and you get a certificate, which is in the loft. He even turned down a knighthood. A knighthood makes you different, doesn't it? And I don't want to be different. Having retired, Sanger is content these days to tend his garden outside Cambridge, though he still makes the occasional self-effacing and cheerful appearance at the Sanger Center, the genome sequencing facility near Cambridge that opened in 1993. Sequencing would confirm one of the most remarkable findings of the 1970s. We already knew that genes were linear chains of A's, T's, G's, and C's, and that these bases were translated 3 at a time in accordance with the genetic code to create the linear chains of amino acids we call proteins. But remarkable research by Richard Roberts, Phil Sharp, and others revealed that in many organisms, genes actually exist in pieces, with the vital coding DNA broken up by chunks of irrelevant DNA. Only once the messenger RNA has been transcribed is the mess sorted out by an editing process that eliminates the irrelevant parts. It would be as though this book contained occasional extraneous paragraphs, apparently tossed in at random, about baseball or the history of the Roman Empire. Wally Gilbert dubbed the intrusive sequences introns, and the ones responsible for actual protein coding, i.e., functionally part of the gene, he named exons. It turns out the introns are principally a feature of sophisticated organisms. They do not appear in bacteria. Some genes are extraordinarily intron-rich. For example, in humans, the gene for blood clotting factor VIII, which may be mutated in people with hemophilia, has 25 introns. Factor VIII is a large protein, some 2,000 amino acids long, but the exons that code for it constitute a mere 4% of the total length of the gene. The remaining 96% of the gene is made up of introns. Why then do introns exist? Obviously, their presence vastly complicates cellular processes, since they always have to be edited out to form the messenger RNA. And that editing seems a tricky business, especially when you consider that a single error in excising an intron from the messenger RNA for, say, clotting factor VIII would likely result in a frame shift mutation that would render the resulting protein useless. One theory holds that these molecular intruders are merely vestigial, an evolutionary heirloom left over from the early days of life on Earth. Still, it remains a much debated issue how introns came to be, and what, if any use, they may have in life's great code. Once we became aware of the general nature of genes and eukaryotes, organisms whose cells contain a compartment, the nucleus, specialized for storing the genetic material, prokaryotes, such as bacteria, lack nuclei, a scientific gold rush was launched. Teams of eager scientists armed with the latest technology raced to be the first to isolate, clone, and characterize key genes. Among the earliest treasures to be found were genes in which mutations give rise to cancers in mammals. Once scientists had completed the DNA sequencing of several well-studied tumor viruses, SV40 for one, they could then pinpoint the exact cancer-causing genes. These genes were capable of transforming normal cells into cells with cancer-like properties, with, for instance, a propensity for the kind of uncontrolled growth and cell division that results in tumors. It was not long until molecular biologists began to isolate genes from human cancer cells, finally confirming that human cancer arises because of changes at the DNA level and not from simple non-genetic accidents of growth, as had been supposed. We found genes that accelerate or promote cancer growth, and we found genes that slow or inhibit it. Like an automobile, a cell, it seems, needs both an accelerator and a brake to function properly. The treasure hunt for genes took over molecular biology. In 1981, Cold Spring Harbor Laboratory started an advanced summer course that taught gene cloning techniques. Molecular Cloning, the lab manual that was developed out of this course, sold more than 80,000 copies over the following 3 years. The first phase of the DNA revolution, 1953 to 72, the early excitement that grew out of the discovery of the double helix and led to the genetic code, eventually involved some 3,000 scientists. But the second phase, inaugurated by recombinant DNA and DNA sequencing technologies, would see those ranks swell 100-fold in little more than a decade. Part of this expansion reflected the birth of a brand new industry, biotechnology. After 1975, DNA was no longer solely the concern of biologists trying to understand the molecular underpinnings of life. The molecule moved beyond the academic cloisters inhabited by white-coated scientists into a very different world, populated largely by men in silk ties and sharp suits. The name Francis Crick had given his home in Cambridge, the Golden Helix, now had a whole new meaning.